Hi, I'm Abby, and today I'm going to be talking a little about my senior research project with the dry bean breeding and computational biology program. When most people think about plant breeding, they probably picture someone walking through a field with a clipboard. And that has historically been true. But today, we're using drones, spectral reflectance, and genetics to try and answer one big question. Can we connect what we see from the sky to what's happening in the DNA of the plant? Dry bean development and productivity are driven by complex physiological processes happening throughout the growing season, things like chlorophyll accumulation, canopy development, and stress response. The problem is, these processes are difficult to measure using traditional phenotyping methods. Visual scoring is subjective, and endpoint measurements miss what happens in between. So, we developed a drone-based phenomics pipeline to capture time-variable data across multiple dry bean market classes. We flew a multispectral drone over our field trials at multiple time points during the season. The drone captures reflectance at different wavelengths, including visible and near-infrared light. From that reflectance data, we calculate vegetation indices like NDVI and GNDVI. These act as indicators for physiological traits, such as canopy vigor, disease or pest presence, and chlorophyll content. So, instead of a single end-of-season measurement, we now have dynamic, quantitative trait data across time. But phenomics alone isn't enough. We want to know whether these phenomic traits are genetically controlled. So we test candidate genetic markers to see if they're associated with the phenomic patterns that we've observed. And this is where the lab work begins. To study genetic markers, we first extract DNA from leaf tissue. Small samples of young leaf material are collected from each genotype. The tissue is ground up to break open plant cells, and a chemical extraction buffer is used to lyse membranes and release DNA. We then purify the DNA, removing proteins, cell debris, and other contaminants until we're left with a relatively clean genomic DNA. That then becomes our template for PCR. PCR, or polymerase chain reaction, is a technique used to amplify a specific DNA region. We first design primers that target the candidate marker region we're interested in, and then, during PCR, the DNA goes through cycles of heating and cooling. First, the DNA is denatured, which means the two strands are separated. Then, primers are annealed to the target region, and a DNA polymerase copies the region. If all goes according to plan, after 30 to 35 cycles, millions of copies of that specific marker region are produced. Next, we run the PCR products on an agarose gel using electrophoresis. DNA fragments are negatively charged, so when an electric current is applied, they migrate through the gel towards the positive electrode. Smaller fragments move faster than larger ones, since they have an easier time migrating through the gel. After the gel is stained, we can visualize the bands under UV light. What we're looking for is the presence or absence of specific DNA fragments, or differences in fragment size, that correspond to particular alleles of a genetic marker. If a band appears at the expected size, it indicates that the genotype carries that marker allele. If the band is absent, the allele is likely also absent. By comparing band patterns across genotypes, we can determine which plants carry which marker variants. This is important because plant breeding relies on selecting the right individuals. If we can use drone-based phenomics to rapidly screen large populations and confirm those signals with validated genetic markers, we can dramatically improve selection accuracy. This means faster selection of superior genotypes, reduced reliance on subjective scoring, earlier selection decisions, and ultimately accelerated genetic gain. We're essentially building a repeatable phenomics-to-genomics pipeline that links reflectance data from the sky to validated genetic markers in the lab. So instead of just asking which plant looks best, we're asking what does the drone see, what does the data say, and what does the DNA confirm? And when those answers align, that's when breeding becomes truly powerful.